Okay, good day, Dr. Jaya. So we are from Group 4 and this is my group members and I'm the leader, Bong Kwani, with the student ID SP22025.
I'm Yong Xuan with SP22030.
I am Liu Xingyi, my matric number is SP22033.
And I am Lim Kim Tong, my SP number is SP22032.
I am Ye Ipei. My matrix ID is SP22031.
I don't need no light to see you shine.
So for question 1, do the primary amino acid sequence of CBS AGC. And this is the amino acid sequence that we have built. For A, identify and describe the N-terminal and C-terminal of this peptide.
We can see from the model, the N-terminal is with the amino group at the left-hand side, and the C-terminal is have the pre-carboxyl group at the right-hand side. Normally, we build the peptide. The N-terminal is on the left-hand side and the C-terminal is on the right-hand side.
For B, show and explain the peptide linkage.
So the peptide bonds join together the primary structure of protein. It is a covalent bond formed when carboxyl group of one molecule reacts with amino group of another molecule. So this is the first peptide bonds formed between the cysteine and valine. The second peptide bonds formed between the valine and serine. The third one is formed between the serine and alanine. And the next peptide bonds formed between the alanine and glycine, and the last is formed between the glycine and cysteine.
For C, show where a disulfide bridge likely to happen and explain its importance.
So the disulfide bridge is likely to happen in is the sulfuridyl group, which is the thiol group, and it will break the bond and form together. This will become the disulfide bridge, and the importance is the disulfide bridge. It can stabilize the tertiary and quaternary form in two different ways, which is the intermolecular disulfide bond and also the intramolecular disulfide bond. That's it.
For D, show where hydrogen bond likely to happen and explain its importance.
The hydrogen bond is formed between the amide group and the carbonyl group, so there are total 4 hydrogen bond formed. This is the first one, hydrogen bond, and the second hydrogen bond, the third hydrogen bond, and the fourth hydrogen bond.
The importance of the hydrogen bond is it will determine the 3D structure and the property that are acquired by the synthetic and natural protein. Besides that, hydrogen bond provides stabilization of protein and make the protein function properly. Okay. For E, is there any difference between this structure to that of CGA, SVC, and Shirley? Okay, so for the amino acid, at first we build is CVS and also AGC. And right now, for the structure of CGA, SVC, the both set of cysteines is remain, and the valine and also the glycine need to be changed. And for the serine and also aniline have to be changed. This is the new amino acid sequence form, which is the cysteine, glycine, alanine, serine, valine, and cysteine. Okay. So, if the sequence of the amino acids are different, it means that their primary structures of this peptide are different. The sequence of the amino acids is essential is because it will give rise to their identity, characteristic, and also their functions. So, as a result, it means that the author in the amino acid sequence will alter their function directly.
For question 2, we are asked to build ribose and deoxyribose sugar. This is the ribose sugar and this is the deoxyribose sugar.
So the difference between the deoxyribose and the ribose sugar is the deoxyribose will lack of one oxygen. So this is the difference between the ribose and the deoxyribose.
And question A is asked to describe using our model the particles of C2-endo and C3-endo for each. We can compare first. In the ribose sugar normally has C2-endo. C2-endo is move the carbon 3 toward to the carbon 5. And the deoxyribose sugar normally has C2-endo. C2-endo will move the carbon 2 to the carbon 5. If compare C3 and C2-endo, the difference is the distance between carbon 3 and carbon 5. C2-endo will have the further distance if compared to the C3-endo. C2-endo is deoxyribose sugar because it has the higher sugar particle energy and one more hydrogen bond with the protein than if compared to the C3-endo does. And followed by the next question is, can a uracil connect to the sugar? Show it.
So this is the uracil that we built. So it seems like this.
And this is the nucleoside. Lastly, we need to build a nucleotide which is add on another phosphate group.
So this is the phosphate group that we built.
After that, this is the complete nucleotide which have the phosphate group, sugar, and also a base.
How to love someone else. I don't know how to forget your face.
For question 3, we need to build an alpha-D-glucose and a beta-D-fructose cyclic structure. So this is the alpha-D-glucose, the structure that we built, and this is the beta-D-fructose we built. For A, describe the beta-D-glucose structure. So this is the structure of the beta-D-glucose. We can saw that in the ring, there are 5 carbon and 1 oxygen. So from the first to the 4th carbon, 1, 2, 3, 4, each carbon linked to 1 hydrogen atom and 1 hydroxyl group. And at the 5th carbon, it linked to the 6th carbon, and the 6th carbon linked to 2 hydrogen atom and 1 hydroxyl group. So we can saw that the 6th carbon is outside of the ring. The difference between the beta-D-glucose and the alpha-D-glucose is the direction of the hydrogen atom and the hydroxyl group at the carbon 1. So in the beta-D-glucose, the hydroxyl group is on the top and the hydrogen atom is on the bottom. While in the alpha-D-glucose, the hydroxyl group is at the bottom while the hydrogen atom is at the top. For B, form a maltose and show its glycosidial linkage and name it. So this is the glycosidial linkage between the maltose. Maltose is formed by 2 alpha-D-glucose. And the glycosidial linkage is called alpha-1,4-glycosidial linkage. Why? It is because at this bond, the C1 carbon linked with the glycosidial bond, and this is the 1st, 2nd, 3rd, 4th linked with the 4th carbon. And thus, because this is formed by two alpha D glucose, so it is alpha 1,4 glycosidic linkage. And for C, explain why it is a reducing sugar.
So for the alpha glucose, this is the presence of the aldehyde group, which is at carbon 1, and it makes them to undergo oxidation readily to form carboxylic acid, and in the process the reactive agent are reduced easily. Whereas for the beta D fructose, is the presence of the keto group, which has the carbon 2. It has the potential to get oxidized through several intermediate tautomeric shifts into an aldehyde and reducing the other compound it reacts with. That's it.
Question 4. Build urea and methylurea structures. Use them to answer these questions.
So this is the structure of urea. Urea, also known as carbamide. Urea consists of 2 amino group and 1 carbonyl group. When urea added with 1 methyl group, it will form methylurea.
A. Which of the substrate above preferred by urease for catalyst?
The substrate is urea. Urease will catalyze the hydrolysis of the urea to form ammonia and carbamic acid. The carbamic acid will further decay to form carbon dioxide and ammonia.
B. Explain the model or hypothesis that describe the action of this enzyme on the substrate.
The model that explains this action is log and key model. The urea will act as the key and the enzyme urease will act as the log. The urease enzyme is absolute specificity, which is very specific, that can only bind with one type of substrate, which is the urea. The substrate urea will bind to the active site of the urease enzyme. After that, they will undergo hydrolysis, which will form the carbamic acid and ammonia. Carbamic acid will then decay to carbon dioxide and ammonia.
Okay, so for part C, is there any another hypothesis for enzyme to substrate interaction? Explain by using your chemistry model for this hypothesis if you possibly can do so by being innovative. So the hypothesis I choose is the induced feed hypothesis, and here is the enzyme we use. This is a part of the adenylate kinase. This is a combination of 2 aspartate and 1 adenine. And here, this is the ATP we use. But because of we don't have the enough of the phosphate group, so we just pretend this is the ATP, the triphosphate. So we use the enzyme, which is act as a log, and also the substrate, which act as a key. And when they do the binding, their shape will change slightly, so that they can alter their shape, and also they can fit completely to each other. The binding of the enzyme and to the substrate will form a byproduct of ADP and phosphate group, and the ATP will further decay or break down to AMP and also another phosphate group. In the end, the final product will be AMP, the adenosine monophosphate, and also 2 phosphate group. So the binding of the phosphate group will just detach like this, and this will become the ADP, the adenosine diphosphate, and also the phosphate group. So this is all for the hypothesis theory.
It's been a long day without you, my friend.